Hello students, welcome to EPG Pathshala again, and I am Dr. M.N. Gupta, Emeritus Professor at IIT Delhi. Today we are going to talk on the module Humoral and Cellular Immunity from the paper again, which is Immunology. Humoral immunity means the protection offered by antibodies. Antibodies themselves are produced by B lymphocytes. Cellular immunity, on the other hand, means the immune mechanism which is mediated by T cells, which are another class of lymphocytes. The following are the objectives of this module. Number 1, to understand how acquired immunity evolved to make innate immunity more versatile, because in the context of the evolution, it is the innate immunity which evolved first and acquired immunity evolved much later. We also will try to understand how innate immunity cooperates with the humoral immunity and the cellular immunity, which of course together constitute acquired immunity. We will also furthermore try to understand briefly what are the respective roles of these two parts of acquired immunity, that is the humoral and cellular immunity. So the concepts basically which are involved in this module is to see that how initially we have innate immunity. This, in terms of the immune response, is followed by continuation of the innate immunity, initiation of the humoral response and cell mediated response. And during this process, towards the later phase, this also leads to creation of the memory cells. So it is the right time to talk of the relative role of the innate immunity and the acquired or adaptive immunity. With regards to the latter, we also need to look at the broad level of how humoral and cell mediated systems cooperate. What actually the immune system achieves? Some protozoan parasites like African trypanosomes live in the blood. Other multicellular parasites like tapeworm live inside the tissues or organs. Many bacteria try colonization of the epithelial surfaces, and if they succeed in overcoming this surface barrier, they also multiply inside the tissues. Some other protozoa, for example the Plasmodium species, which cause malaria, and Trypanosoma cruzi, which causes Chagas disease, these have intracellular phases. Viruses also enter cells to multiply. Whether the pathogen is inside the cell or outside the cell is important. To destroy the microbe inside the cell, humoral response, that is the antibody mediated response, does not work. What we need in such cases is the cell mediated immune response. This is important to keep in mind to appreciate the evolutionary need of the both arms of acquired immunity. The pathogens which cause diseases are very diverse in nature. They could be viruses, bacteria, fungi, protozoa and helminths, that is the worms. In viruses themselves, viruses can be either DNA viruses or RNA virus. Each virus is associated with the disease which its infection causes. This slide shows some of the diseases which are caused by DNA viruses like adenoviruses, herpes viruses, pox viruses, papaviruses, etc. The disease caused in humans by these DNA viruses are also listed here. Similarly, there is a list of RNA viruses, and the diseases which are caused in humans by these RNA viruses consist of influenza, mumps, measles. Common cold viruses are also RNA viruses. Polio virus is also an RNA viruses. Rubella, rabies, these are also caused by some RNA viruses which are mentioned here. Let us also look at illustrative examples of the bacteria, fungi, protozoa and worms which cause their own set of diseases. Different pathogens are known for each type of microorganisms or virus. In bacteria, we have a gram-positive cocci like staphylococci or streptococci. We also have a gram-negative cocci, and then we have a gram-positive bacilli and gram-negative bacilli. We also have anaerobic bacteria like Clostridia. So they are of diverse nature. Each bacteria is known to cause corresponding diseases which are listed here. Fungi also causes many diseases which are listed here. Protozoa also are very difficult infections to take care, but their immune mechanisms which initially ensure that the system is protected against them. Worms can be intestinal worms. They can invade other tissues. There are certain worms which infect blood or liver. Many of the diseases which are caused by the worms are identified and are shown here in this table. So the immune system also faces many challenges. First of all, the diversity of the pathogens have to be meshed by the immune mechanisms. Then there is an issue of the life cycle of the pathogens, and their location in the body has to be countered by these mechanisms. So the logistics have to cover both space and the time frames. Many pathogens mutate, some very rapidly. So in such cases, these become actually the evading tactics for these pathogens. They try to evade the reaction of the immune system, and the immune system has to be geared towards so that these evading tactics have to be taken care of. The invasion by pathogens can be in the extracellular or intracellular space. This is shown in this table. It may be added that to a varying degree, nearly all pathogens do have an extracellular phase. The kind of protective immunity with each kind of infection evokes is a mix of both innate and adaptive or acquired immunity. As we can see from this table, the extracellular spaces are either interstitial spaces, blood, lymph, and then we have epithelial surfaces. Among the intracellular space, we can talk of the cytoplasmic kind or the vesicular kinds. The organisms which normally take part in these kinds of infections are given. The protective immunity which they provoke is also shown against each kind of infection. It should be made clear at this point that a particular infection provokes different arms of the immune system. Let us take the example of the encapsulated gram-positive cocci, which are major examples of bacteria which can resist phagocytosis because of the protective action of the polysaccharide capsules. However, opsonization by specific antibody, which is a part of the humoral immune response, or by complement, which is a part of the innate immunity, facilitates their phagocytosis. By opsonization, we mean coating these pathogenic cells so that their phagocytosis is facilitated. What we see here is a clear illustration of the fact that we actually need multiple arms of the immune system to take care of the infecting organism. This can also be seen by taking up the example of the viruses. Viruses can be neutralized by antibodies. Production of such antibodies depend upon the cells of the immune system, which are called Th2 cells. This is of course a part of the cell mediated immunity. Once inside, Tc cells kill the infected cells. Intravesicular pathogens infect macrophages, which are helped to destroy these by Th1 cells, which are of course also part of the cell mediated immunity. So what we see here is that again different arms of the immune system come into play. This again is aimed to emphasize that there is a need for both innate and acquired immunity. The way different organisms cause tissue damage also differs. Many extracellular microbes act by releasing toxins. Neutralizing antibodies are induced and try to overcome this effect. Intracellular pathogens infect cells. T cells not only destroy the damaged cells, it thereby prevents the spread of viral infection. In some cases, this itself leads to pathological situations, though. The site of infection is important. Streptococcus pneumoniae in the lung causes pneumonia. If the organism infects blood, it leads rapidly to total systemic failure. The immune response in many cases can be localized. For Vibrio cholera, the intestinal pathogen, mucosal system of the gastrointestinal tract uses acquired immunity mechanism to respond. One general feature is that the innate immunity comes into play first. If it is overcome, acquired immunity takes over. The latter also takes effect after several days. Immune responses, as we will see in more detail, are actually well orchestrated phenomenon. The level of infectious agent increases as the pathogen replicates. When number of pathogen exceed the threshold dose of antigen required for an adaptive response, the response is initiated. The pathogen continues to grow, retarded only by the innate and non-adaptive responses. At this stage, immunological memory also starts to be induced. After 4-5 days, effector cells and molecules of the adaptive response starts to clear the infection. When the infection is cleared and the dose of antigen falls below the response threshold, the response ceases. However, the antibody, residual effector cells, and also immunological memory provide lasting protection against reinfection in most cases. Let us discuss first of all the timeline of the infection. First of all, the infection has to overcome physical, chemical and biological barriers of entry. Innate immunity fights the colonization of the pathogen. This includes local inflammation and cytokines inducing other effector cells and molecules to gather at the site of infection. Even these innate immune responses distinguish between self and non-self surfaces through a number of germline coded receptors. At best, these innate immune mechanisms are able to deal with the infection. There are few deficiency diseases related to these non-adaptive or innate immune responses which are known. So we never know the success rate of these mechanisms. At worst, these innate immune mechanisms are unable to contain the infection. If the infection reaches a threshold level, adaptive immune responses get initiated. Again, in this period and even subsequent time period, innate immunity, first alone and then in cooperation with acquired immunity, fights the infection. This infection is either contained by the lymphoid tissue, by the immune mechanisms. If the infection overwhelms these, then it spreads through blood. In both phases, humoral and cell-mediated immunity participate together. Once the level of infection falls below the threshold required to operate acquired immunity, the effector cells and the memory cells prolong the protection, as we have mentioned. In this last phase of infection clearance, humoral response clears the extracellular infectious particles and cell-mediated immunity clears the residual intracellular infection. At the end, the consequences are that no residual sign of infection remains, some tissue damage is caused, infection is contained but remains in the latent form, in some cases, for example, by Mycobacterium tuberculosis. If the acquired immune responses are severely compromised, such as in AIDS, the disease reappears as systemic infection. It is necessary to compare the time course of infection in normal and immunodeficient animals. Here, the red curve shows the rapid growth of microorganisms in the absence of innate immunity when macrophages and polymorph nuclear leukocytes are lacking. The green curve shows the course of infection in mice and humans that have innate immunity but have no T or B lymphocytes and so lack adaptive immunity. The yellow curve shows the normal course of infection in immunocompetent mice or humans. The innate immunity response is essential for initiation of acquired immune response. To start with, in the absence of innate immunity, the level of infection can rise unchecked and very rapidly. If innate immunity was the only arm of the immune mechanisms, It will not be able to deal with any infection at a serious level. It will fight for a while, try to contain it, and then the infection will overcome it. It is the time period around which acquired immunity comes into play and contains the infection. The innate immunity first of all creates responses around the local environment of infection. In the case of bacterial infection, inflammation is the first visible response. The macrophages residing locally have toll-like receptors through which they recognize the lipopolysaccharide coat of the bacteria. The cytokines and chemokines are released by the macrophages and bring in PMN leukocytes such as neutrophils and monocytes. So far, this is all relatively non-specific in nature. The second important phase is the activation of the local dendritic cells. These dendritic cells ingest the antigens and move from the lymph in the infected tissue to enter the secondary lymphoid tissues. At this point, the fight against infection moves beyond the local site of infection. It is here that the naive T cells come into play. This is the antigen specific response. The first response is the cell mediated in this case. Clonally selected T cells enter the circulation and can reach back the local site of infection. Here what we see is the traffic and recirculation of lymphocytes through encapsulated lymphocyte tissue and the site of inflammation. It is in the lymphoid tissue that these activated T cells run into B cells. Clonally selected B cells release a specific antibody. Humoral response thus initiated further ensures containment of the infection. Antibodies play the role of an adapter molecule in many ways. When a microorganism lacks the inherent ability to activate complement or bind to phagocytes, the body provides antibodies as flexible adapter molecules. The body can make several million different antibodies able to recognize a wide variety of infectious agents. Thus, the antibody illustrated binds microbe 1 but not microbe 2 by its antigen binding portion FAB. The FC portion may activate complement or bind to FC receptors on host cells, particularly phagocytes. Phagocytes have an intrinsic ability to bind directly to bacteria and other microorganisms. This is much enhanced if the bacteria have activated complement. They will then have bound C3b, which is a complement molecule, so that the cells can bind bacteria via C3b receptors. Organisms which do not activate complement well, if at all, are opsonized by antibody, which can bind to the Fc receptor on the phagocyte. Antibody can also activate complement, and if both antibody and C3b opsonize the microbe, binding is greatly enhanced. The design of the antibody as an adapter molecule has to be appreciated. Its FAB part has specific recognition tailored to these diverse antigenic substances. That is why this is fashioned out of the variable part of the molecule. This is where the humoral response comes in. The other part relates to the interaction with the components of innate immunity. These interactions are common. That is, these are not dependent upon the specific antigen which is bound to FAB. Hence, these do not need tailoring of any antigen. This structural part is thus made up of constant region of the antibody. At the same time, antibody during evolution allowed a strategy where evolution did not discard innate immunity. Antibody is the means by which evolution neatly segued the humoral response to the existing innate immunity. Antibody mediates the classical complement pathway. which is in a way slightly a misnomer since it evolved later than the alternative pathway, but was discovered earlier. That is another route to complement activation, which requires antibody presence and thus differs from the earlier evolved pathway, which could act and does act in the absence of antibody. This is in line with the above picture about the evolutionary progress. So, classical pathway broadened the scope of action by the complement of pathogens which otherwise escaped innate immunity. Many C3b coated pathogens bind to the phagocytes but are unable to initiate phagocytosis, presumably due to weak binding. The antibody provides stronger binding by acting as the adapter. This is confirmed by the fact that a single antibody molecule is not enough. This adapter action, which cross-links the pathogen and the phagocyte, requires multiple linkages. So, either few Ig molecules have to bind close to each other on the pathogen cell surface, or multivalent IgM is necessary. These facts have been actually experimentally verified. In cases where C3b opsonization does not trigger phagocytosis, adding antibodies initiates phagocytic action. The comparison of the low affinity single bond and high avidity multiple bond is illustrated in these figures. How did the acquired or adaptive immunity evolve? Adaptive immunity, as we know now, has two arms. One is the antibody mediated humoral response, and the second is the cell mediated immunity, which is basically involves action by the two kinds of the lymphocytes, the B lymphocytes and the T lymphocytes. As far as the structure of immunoglobulin G is concerned, it strongly suggests that it evolved from a single domain polypeptide. That itself must have evolved from a pre-existing molecule of what we call the immunoglobulin supergene family. The immunoglobulin superfamily is shown in this figure. As we can see, T cell receptor, the CD8, CD4, HLA class 1, HLA class 2 are all part of the immunoglobulin superfamily. Where did the receptor molecule which later evolved into IgG initially get expressed? It may have been a cell similar to cells involved in innate immunity, which later evolved into B lymphocytes, as we know now. All jawed fish have the capacity for adaptive immune response. Jawless vertebrates, hagfish and lampreys do not have adaptive immune response. Cartilaginous fish, the earliest jawed fish, have all the elements of adaptive immune response of both kinds, but in a primitive form. All metazoan have cell surface recognition systems which are capable of distinguishing between self and non-self. This distinction between the self and non-self, as we have seen, is actually a hallmark of the immune action. Let us look at these evolutionary aspects a little further. The Hawaiian sponge Calispongia diffusa is a very primitive metazoan which has three fundamental traits of the immune system of the vertebrates. Its self and non-self recognition systems operate via cell surface molecules. non-self are destroyed by some effector mechanisms. Individuals within a given species have distinct self markers. Juxtaposition of 900 pairs of genetically different Calispongia have exhibited incompatibility, with a zone of dead cells separating the tissue and indicating rejection. Higher invertebrates such as colonial Ascidian Botrytis also has similar self-non-self recognition mechanisms. It is believed that gene rearrangement has been the crucial step in evolution. Not all animals carry gene arrangement to create diversity of antibodies in similar way. In chicken and rabbits, as we will see, the recombined V gene is diversified in bursa or intestinal lymphoid organ respectively. In carcharodon sharks, multiple rearranged VL segments occur in the germline itself. So students, let's now summarize what we have learnt in this module. First of all, we saw that how innate immunity initially tries to contain the infection. It is the persistence of the infection which leads to the acquired immune response. Both antibody mediated and cell mediated immunity are necessary and play a complementary role. It is likely that during evolution, innate immunity existed and later on acquired immunity evolved. Simultaneously, a synergy between the two also evolved. Nevertheless, it should be made clear that the innate immunity is not merely a remnant of the early days of the evolution of the immune system. It is essential for acquired immunity to work. What is more, both kinds of acquired immunity responses work together and in cooperation with the innate immunity. So, immune system can be thought of as a great model for teamwork by cells and molecules of this entire team. Thank you.